Hello, everybody. I am Emily Leproust. I am the CEO of Twist Bioscience. I'd like to welcome you to Writing the Future of Infectious Disease. First, I'll start by saying that I will be making some forward-looking statement today. And at Twist, we are very excited because we write DNA from scratch. And DNA is the most important molecule of the 21st century that is changing the world for the better. Our customers use our DNA in many different areas, in the production of chemicals in a way that's more sustainable by fermentation of sugar, for improving food security, for instance, by delivering fertilizer at the root of plants using engineered bacteria instead of using fertilizer made from fossil fuel. Our DNA is used to develop a lot of new biological drugs. It's used in the diagnostics of disease and virus, like we'll talk about today, and you can even use DNA to store data for the long term. And at the center of everything we do is our technology, where we write DNA on silicon, and the chemistry of writing DNA is known, but what we've done is we've miniaturized that chemistry on silicon. We use very small volume, and thanks to that miniaturization, we get two things. One, we get much higher throughput, so we can make more DNA than anybody else. And two, we can lower the cost of writing the DNA. And of course, we try to pass that cost saving along to you. And based on that technology, we've built a number of products. We can make genes, we have oligo pools, we can make protein libraries, we have NGS tools, and we even have IgG antibodies for biopharma. So in the next few slides, I'll talk about what we do in particular against infectious disease. And I'll take the example of COVID because like many others in the biotechnology area and the research area, COVID has shifted the plan of what we do at Twist. And so we've gone into the fight and we've adjusted what we could do to help in the fight of the global pandemic. And so today I'll present the unique approach that we've done in the fight based on the silicon platform to write DNA. So first we have NGS Panels. And so the idea with NGS Panels is that to detect SARS-CoV-2, you can use RT-PCR, and that gives you a yes or no answer. But we've made NGS Panels where we made DNA probes against SARS-CoV-2 such that you can enrich the virus out of a human sample. And then we can sequence, and that gives you the full sequence of the virus, all 29,000 bases. And you can see that even at very low virus copy numbers, we're able to get sequencing of the virus. And that's quite useful to understand variants, to understand evolution of strains, evolution of lineages between strains. Next, I'm very happy to say that NGS panel has actually gotten FDA authorization, and that has been authorized along with our partner Bioscience. With also beyond just the SARS-CoV-2 panel, we have a respiratory panel. So there, instead of having just COVID, we can detect 29 common human respiratory viruses all at the same time, which can be useful in detecting co-infection. So again, that's an NGS panel. And then the next step beyond that is to look at the whole pan-viral area, where we can detect 3,153 viruses all at once, which is very useful to analyze samples where the virus is unknown. Next, we also have done synthetic control. So for the development of NGS tests, for the development of RT-PCR tests against COVID, you need a positive control. And growing the virus and extracting it is actually quite dangerous. So again, using our silicon platform, we've made COVID in a tube. It's the entire 29,000 base pair, all RNA sequences, and it's cut in 5 pieces. So it's safe. You can brush your teeth with it. Don't do it, but you could. And since as there's been more variants coming out, we have launched more synthetic controls. So now on the web, we have more than 15 different controls for SARS-CoV-2. And then last but not least, we've been able to use the silicon platform that we have to make large libraries of antibodies. And then we've been able to pan those antibodies against the N protein and the S protein of COVID, and we've been able to find antibodies that are neutralizing against those the spike protein. And I'll share with you some preclinical in vivo data. We have used an hamster model where COVID is given to an hamster, and you can see in red that when there is no thing given to the hamster, the weight loss is observed and ultimately death of the animal. But with twist antibodies, especially TB202-3, even at 1 milligram per kilogram, which is a very low dose, you can see that it's quite potent in making sure that there is no weight loss and that the animal survive. And so this demonstrates the highly potent neutralizing effects of our antibodies, and we're working with partners to bring these antibodies to the market to help in the fight against global pandemic. And so to conclude, I hope that you've seen that there is multiple ways that we've followed to address the current challenge facing humanity from infectious diseases. We've been used our platform to build DNA tools that are helpful to you, the scientist, and we are there. We're working for you, and we're there to enable you to do your great work. So with that, I want to thank you very much, and I very much look forward to work with you to write the future. Thank you very much.